So today, Dr. Avnika will give a talk on Drosopho-nanotoxicity. Now, this sounds very fascinating, and I believe everyone would be very eager to know what this Drosopho-nanotoxicity is. So, Avnika, a very warm welcome to you. The forum is all yours.
Thank you, and thank you for inviting me for this webinar. And I really appreciate that you have started with this webinar, especially at the time of pandemic where students are at home and they really need these webinars to update their knowledge. And I will share the screen now. Okay, to begin with, the topic is Drosophila-nanotoxicity. So we will be talking about Drosophila. We will be talking about a bit of nanotechnology, and I will emphasize why we want to study about nanotoxicity as well. So the topic is understanding nano-biointeraction of nanoparticles using Drosophila melanogaster as model organism. So to begin with, first I will tell about what is nanotechnology. When it comes to nanotechnology, we talk about very small particles that is of a size of 10 to the power -9 of a meter. And what is the importance of this is when we go down to this small size, the property of the matter changes and all the physics is being governed by quantum mechanics, and which leads to increases the surface to the volume ratio, leading to novel properties of materials. And the idea was given by Dr. Richard Feynman in his famous talk, There is Plenty of Room at the Bottom. And with his idea being taken up by the various scientists and researchers, we have nano-based application in various fields. For example, in case of medicines, in case of drug delivery, and moving away from medicines, we have nano-based applications even in our day-to-day life. For example, food nanotechnology, that is the usage of nanomaterials in food packages. So as we are moving ahead with nanotechnology, from high-end technology to day-to-day basic products, there is probably a chance of exposure to these nanoparticles unintentionally or intentionally. So perhaps we have overdid the nano part and underdid the bio part. And it is time for us to move from the nano part and understand the bio part. And it is important for us to know what exactly we are dealing with. So taking it forward, we have come up with a field called nanotoxicology, which emphasizes on study of ultrafine particles, that is nanoparticles. And when in the atmosphere, how we can come in contact with this particle is through various routes, that is ingestion, either we can engulf these particles, dermal absorption, that is through the skin, inhalation, that these particles can be inhaled through the air, and these particles can also hijack the olfactory route and reach our brain cells. So the common toxicity known to be induced by these particles is they can cause oxidative stress, they can cause protein coordination complex, they can cause Trojan horse effect and genotoxicity. So since these are charged particles, they can increase the oxidative stress. They can interact with protein molecules, forming a layer over the protein molecule. Like proteins can come and attach to these nanoparticles, forming a protein coronal. And the Trojan horse effect is the word derived from the famous story of Troy, where the Trojan army tried to invade into the city by using a horse statue. Similarly, these particles, they can engulf in the lysosome, and once they are inside the cell, they burst open and they release the ionic particulate form of these particles, causing more toxicity. Moving further, they can also cause genotoxicity directly or indirectly. Either they can go straight to the nucleus, causing genotoxicity, or indirectly by increasing the ROS content of the cell. So these are the known toxicity mechanism for these particles. But the big question is that on long run, how these particles are going to affect, and how far a one-time exposure of these particles can go, and how their lethal effects can be carried forward to future generation. So in order to answer this, we have come to the acceptance of Drosophila as a model organism for studying nano toxicity. And Drosophila has many importance, like it has a complex behavior. We have few ethical concerns. It has complete genome sequence, which has similarity with humans. But the main importance why we want to use these particles in our study to study nanotoxicity is because we want to screen a large number of population, and we can go down to subsequent generation and numbers of time in a very less time. So that advantage can be easily we can achieve through this model organism, that we can screen large number of population, and in a very less time we can get the data of subsequent generation. So these are some of the research papers I have highlighted here, which shows the importance of using Drosophila model, and its well acceptance in the scientific society for studying toxicology. Now, based on this, the aim of our research was to study the toxic effects of two nanoparticles, zinc oxide and aluminum oxide nanoparticles. And we have classified our research into two phases, like phase 1 and phase 2. So phase 1 include various assays through which we can classify these particles as toxic or non-toxic. And the phase 2 will include to understand the mechanism of toxicity, that what could be the probable cellular mechanism causing toxicity of these nanoparticles. So as I mentioned, that we have used Drosophila melanogaster model organism, and how we use these model organism. We culture these flies in a vial of 20 to 50 ml, and we culture them a solid food, which is of high media, mainly consisting of corn flour, yeast, and peptone. And we culture flies at 25 degrees centigrade temperature at a humidity of 55 to 60%, with 12 hours day and night cycle. And the fly strain which we use is Oregon R wild type. So this is one of a very important slide, and it is one of the prior experiment. I would say this is a very basic step of our experiment, but at the same time, it is very important that before our exposure is started, we are able to determine the dose at which we are exposing the flies to, and characterize these nanoparticles that they are in the nano form. So there are various ways through which we can expose flies to nanoparticles, and like inhalation and ingestion. And in our experiment, we have used ingestion method to expose flies to these nanoparticles. So first, to determine the dose, we expose flies to a dose range of 0.1 to 10 millimolar and more than that. And at every stage of fly cycle, we see that either the eggs are given to turn into pupa and larvae, and then larvae are able to pupate and to go up to fly stage. So we have found that 10 millimolar is the most lethal stage, above which these flies are not able to the pupa is not able to pupate into an adult fly. So we determine 0.1 to 10 millimolar range as a safe dose for studying the effect of these particles. Apart from this, this is very important to know that the suspension in which we are preparing the nanoparticles is the nanoparticles are not agglomerating to form a bulk form, and then they are in the nano state. So for that, before you expose flies to these particles, you characterize two things, that is the size and zeta potential of these particles. And this is done by the DLS, dynamic light scattering, and this is done by Malvern DLS machine. So first, we know the size of the particle is within the range of the nanoform. Second, we should ensure that the zeta potential is within the range of good stability, to ensure that agglomeration is not being formed prior to exposure or during the exposure time. Also, since we are adding these particles at desired concentration to the Drosophila diet, it is important for us that also in the diet they are in the nano form. So for that, we have done TEM analysis, and we take 1 microliter aliquot of these of the food containing the nanoparticle, and we place the food on this TEM 300 mesh carbon coated grid. And as you can see in this images, we can see these tiny dots, which are 50, 20 to 30 nanometers zinc oxide nanoparticles. So once you are sure that your suspension and dispersion is perfectly okay in the liquid form, also in the food, and you can also check that once being engulfed by the flies, these particles are able to cross the gut and reach the hemolymph, which is a circulating system. So we also check in the hemolymph, where we can see the nanoparticles, which ensures that these nanoparticles have been engulfed by the flies and it is within the biological system. So in order to assess nanotoxicity of these particles, we do various assays to find it out that these are toxic or non-toxic. So one of them is the climbing assay. In this assay, we place flies in empty tubes, and then we leave them to get acclimatized to this new environment for, say, 15 minutes. And after that, what we do, the flies have basic nature that they climb up to the top of the tube. So we tap the tubes, and most of all the flies, they fell to the bottom of the tube. And once you stop tapping, they run to the top of the tube. So we record this entire procedure, and it's roughly around a 1-minute video. And later on, you analyze this, and you see, as you can see in this picture, that the control flies are able to reach the top of the tube, whereas flies exposed to nanoparticles, they are not able to reach the top of the tubes. So this assay will help us to understand the motor activity and the basal behavior of flies flying. That is it being affected by zinc oxide nanoparticle exposure or any other nanoparticle exposure. Moving on further, we can also look for longevity assay. That is, we can assess that is these nanoparticles affecting the lifespan of the flies. So in this assay, we culture flies in food containing nanoparticles, and we transfer flies to fresh tubes every second day. And each time when we transfer the flies to the new tubes, we count the number of dead and live flies. And this helps us to understand that is there any delay or is there any earlier death in case of flies, which help us to understand that is longevity being affected by these nanoparticles. Now, in order to know any other mechanism which could be affected by the nanoparticle exposure, we can do for the ROS, the DCFD assay, and for the DNA damage, we can do the terminal link tunnel assay. And in our experiment, we chose hemocytes as the test sample. Why? Because these are blood cells and they're circulating. So we can easily assess that to various organs, these particles being transmitted, and on a larger level, what damage they are causing to. So we can see in our study, that is in case of zinc oxide nanoparticles exposure, these nanoparticles have increased the ROS generation in case of flies exposed to nanoparticles. And also in case of tunnel assay, we can see that there is increase in fluorescence of damaged cells when they are exposed to nanoparticles. Now, this is one of the very interesting and I would say colorful assay. And this is two-taste tasting assay, which help us to know the cognitive behavior of flies. So what we do in this assay, we expose flies to a sucrose diet or a water containing diet, and we culture flies on this diet for 2 hours. And after that, we starve flies for 8 hours. And then we give them a choice of food. This is a 96 well plate where you have food labeled with 2 colored dye, that is blue and red. And you can, accordingly, you can choose like blue with sugar or red with water, and vice versa, because vice versa. And this experiment is done in closure in dark environment, so that there's no interference of light. And after 2 2 hours exposure of where flies can select between the 2 different diets. We freeze thaw the flies, and how we can know that which fly has the indulged blue or red food is that we can clearly see that they have the color in the abdomen. So we can study different stages we can study different stages we can study different stages of development. So in case of larvae, we can see we can do this assay, which is a crawling assay, where we place a fly at the center of the tube. So we place a larvae at the center of the tube, and the larvae will move in a straight line. So in case of nanoparticle exposure, we can see that there is a lot of twist and turn, and the larvae is not moving in a straight line. However, in case of control, we can see that they have a straight trajectory. So this behavior was also affected by on exposure to nanoparticles. We can study various forms of toxicity when we are using Drosophila, genotoxicity, then neurotoxicity, and for immunotoxicity, this is a very simple experiment where you can take larvae periods and then you boil it to 70 degrees centigrade temperature, and we have crystals when they're exposed to such a high temperature, they burst open, leading to melanin production. So we can observe that in form of tiny black dots in the abdominal region, and we can count these dots and know that is exposure to nanoparticles have affected any immunogenic response. Now, one of the main finding of this study is phenotypic screening. So we can screen number of flies through various parameters, like we can observe the wings, eyes, thorax, body color, and other phenotypic changes. And in case of zinc oxide nanoparticles, we have observed we have deformed thorax and single wing flies study. And here also you can see that the thorax failed to develop and you have a single wing fly. You have a deformed wing here. And as we move further to subsequent generation, this deformity increases. In case of aluminium oxide nanoparticle exposure, we have observed flies which are with 5 legs and sometimes 4 legs. And if there is all the appendages are there, then there are deformities in the appendages. You can see the abnormal wing phenotype. We have a wing blister here, and then this is a deformed leg. And also we have observed renal failure in these flies on exposure to nanoparticles. You can see the swollen abdomen of a fly here showing us renal failure, which could be mainly because of accumulation of these particles in the Malpighian tubes. So just by observing the flies under the microscope, phenotypically, we can clearly see that how these nanoparticles have affected on parent flies as well as in subsequent generation. So we can know on longer run how these exposure to these particles have affected flies in a longer run. Checked in subsequent generation by different cross. If 2 mutant flies are cultured on normal diet, how the and a normal diet, how the progeny is being like to know the lethality of the damage caused by these nanoparticles. And it has been observed when 2 mutant fly has been cultured on a normal diet, they fail to the eggs were laid and then they turn into larvae and pupa, but the pupa failed to turn into an adult fly. And even when the abnormal flies have been grown on normal diet, in the progeny, we have flies which are abnormal without any nanoparticle exposure. So this means that once exposed, these nanoparticles are causing damage in further subsequent generations. So far we know that these particles are toxic, they are causing damage in like phenotypic changes are there in the flies, there's changes in behavior of the flies. What we don't know is what could be the mechanism of toxicity inside the cell, what is happening which could be leading to these phenotypic changes. So in order to understand that, we adopt two approaches. One is to know the global proteomics, that is toxicoproteomics approach. And the second is, since the zinc phenotype is very similar to the wingless protein phenotype, so we protein in the wing disc. Now, in case of flies at the developmental stage, that is larvae, we have imaginal disc for all the this and antennal disc. So in case, since we have deformities in the wings, so we picked up the wing disc and study the expression of wingless protein in the wing disc of flies. So toxicoproteomics is being mainly done by LC-MS-MS studies, and we do LC-MS-MS of control and mutant flies. And once we studied the data further for gene ontology through Flugo, string, and cytoscope, and the expression of the proteins which are only expressed in the control and the test through strap software, we found out that the mainly proteins related to the cytoskeleton, which is like actin, laminin proteins, they are being getting affected on exposure to these nanoparticles. So somewhere they're going and interacting with these particular proteins and affecting the downstream expression. When we have studied the expression of wingless proteins, we have observed that in case of nanoparticle exposure, the expression of wingless protein is being deviated. Now, wingless is similar to the wing protein of humans, which regulate stabilization of beta-catenin in the nucleus and actin proteins outside the nucleus. So based on these studies, we can possibly say that these nanoparticles, they interact with the Wnt pathway, and later on they are affecting the expression of cytoskeleton protein. They're stabilizing the cytoskeleton proteins, leading to the phenotypic changes which we have observed in case of our study, that is a single wing flies. Also in case of aluminium, we have observed flies with certain phenotypic changes, and mainly in that study as well, the cytoskeleton expression of cytoskeleton protein is being altered, leading us to the conclusion that these exposure to these nanoparticles are affecting the cytoskeleton proteins. Now, the outcome of this study is that we have a better understanding of these nanoparticles, and we can easily know that these are toxic or non-toxic. And all this data is adding to the future regulatory measures. They are adding to the regulatory measures for better sustainable society. And currently we are in a situation, we have two groups. One is we have nano-based applications, and the other is no-nano. So we need to talk, and there should be a safety guideline laid down by a certain way to in between the two, where we can still use nanotechnology. We're not saying that these are bad particles, but we should understand the mechanism where how they can interfere with the biological system for leading to better application and safer application of these particles. And I would like to end with a famous saying of my favorite movie, Lucy, that is, ignorance doesn't bring chaos, but knowledge, not knowledge. So that is what we are looking forward, is that knowledge related to these particles and in detail that how they will affect with the biological system. And these are some of my abstracts and publications related to this particular study. And these are some of the other publications. And this is our team, and the whole work is being carried out under the supervision of my guide, Dr. Ekta Kohli. I would also like to thank Dr. Deepthi and Prasad, which is the HOD of our department, and the lab members, Mitab sir, Dolly, Neha ma'am, and my colleagues, Rahul and Kalyan.
Dr. Avnika, it was a wonderful talk.
Thank you. Thank you.
It was a wonderful talk, and it was really pleasure listening to you. I guess whoever is working in the field of Drosophila, they can definitely look up to you as a person who can help them in designing their research work. And also, I think some wonderful images also I saw. So you can also be like you can say a first-hand experience you can share with them. So as far as the Drosophila model is there, how easy it is to work with the Drosophila model? How easy or difficult it is?
I would say to begin with, it will be little difficult because you are dealing with a very tiny insect, but microscopes can solve the issue. But once in practice, it become easier and easier. And if you see that people have easily studied the brain and small cells and tissues, so it is not a difficult task to study flies in the current scenario. And I would also like to mention that in my slide, I've just mentioned about genotoxicity, but there are like, as it is, I mentioned in one slide of neurotoxicity. So it is like a big spectrum where you can study the effect of these particles in brain and on the immune system. And I would say also the cardiotoxicity of these particles, which is, I would say, yet an area to be studied further.
Okay, okay. So I think these days people are working on neurology on various aspects. Like for example, your lab, I have seen they work on ommatidia that I have seen closely. So in this, I would say if you're working on microtubules, so in studies of microtubules, how easy like if you pick up a nanotoxicity or nanoparticles for that, so how easy is it? Is it feasible or how difficult it is?
I think it is quite feasible because you have online procedures for that, and it is quite feasible to go for such studies in case of flies. I would also like to mention that there are a lot of, because we have not talked about the Gulfo US system in flies in my slides, but we have a well-established Gulfo US system where we have transgenic flies for different gene and particular proteins. So in which you have like two flies, and in one you have the US system, the other you have gene of interest, and you can also generate those transgenic flies very easily. So if you want to study any protein or gene in specific, that could easily be done with that help of Gulfo US system, and you have that expression of that particular gene in a certain, let's say, I would say, suppose the eye of fly. So you just by looking at the eye morphology, you can study the expression of particular gene or protein. So that makes the Gulfo US system, that makes more easier the studies for certain case study tests.
Okay, okay. So I have read somewhere that there are some potential like side effects of iron oxide nanoparticles. Is it true?
There are, as I mentioned, it is not the scenario that we are saying that these particles are bad particles or there are side effects. What we are aiming is the better understanding of how these particles interact with the biological system. So the more we are clear on that, it will be easier for us to target those particles in certain applications. So then side effects and the toxicology part will narrow down if we know the exact mechanism, and maybe we can code them with some thing, or we are coming up with like green nanoparticles or safe coatings. So we can still use this particles, but on the safer side. So the better we know, the safer it will be.
Okay, okay. So recently there are also some work going on nanofertilizers. You have any update on that?
Yes, I have heard about that nanofertilizers, and there are also toxicity studies about nanofertilizers. Because
Hmm.
recently in my work have gone through this nanoplastics and nanofertilizers and the toxic effect of both. So this is also another area of research where we can go for this.
Okay, okay. Sounds really exciting because we have some old concept of fertilizers and how nanofertilizers are going to be a game changer. That is something which you have to see.
Yes,
Okay, so
and also again,
yeah, please.
but again, it would be like we should know in detail how they will interact with the plant system and their
Hmm.
metabolism and how long they will be there in the plant. Because of the size, they can easily reach up to the cell organelles and other things. So it is very important for us to know that retention is there and how, for how long, and if it is there, then what could be the
Okay. Uh, Avnika, one last question. So, application of the adsorbent property of nanoparticles is something which people have been exploiting time and again. So, recently, people are using this property as like fighting against pollution. So,
Mhm.
you have anything to say or any comment regarding that?
Not, I would not, I'm not aware about the pollution exactly, but I have seen certain papers and videos as well where they have nano paints
Right.
which applications are observed. But as far as pollution is concerned, I'm not very sure about that, what exactly it would be.
Okay. Okay. That's fair enough.
But it is an interesting topics. I will certainly look up to it that what all the applications are where they're using it.
Right, right. I think these days research has also become very fascinating. So you pick up a topic which is very fascinating, and then you can tell your friends, "See, I'm working on nanofertilizers." Yeah,
Yes, and it's always like when you read papers or where you are in the, you catch up to like, oh, this is an
Mhm.
exciting topic to look forward to.
Okay. So before we end this session, any words for the Bioimaginee platform or any scope where we can improve?
No, it is perfectly fine. I will give it a thumbs up. And as I mentioned already, that I really appreciate that you have started with this, especially in certain these timings where we have pandemic and we have lockdown. So it is very difficult to come up to the interactive life science or scientific platform. So it is a very good beginning, I would say, and all the best to you.
Thank you, thank you. I think with the support of all of you like this, I can definitely look forward to take this platform to a bigger stage where more and more researchers can, my idea is like more researchers may connect to each other and their work become easier. That's it. So with this, I really thank you for your time and thank you for the wonderful talk. And there were some questions which you have answered already. In case somebody wants to, so especially to the viewers, somebody wants to contact Dr. Avnika, you can always write in the comment box, and I believe she will be always like helping you out in all possible ways as far as your research is concerned. So with this, I end today's session. Thank you everyone for attending it. Thank you, Dr. Avnika, for the wonderful talk. Thank you.